Okay, so the next step in this process is the cellular RNA polymerase is going to transcribe the viral mRNAs from the cccDNA. So we use cellular RNA polymerase, abbreviated polymerase II. All right, and then after that, these are going to be translated into the proteins that will form the nucleocapsid eventually. All right, so back on this side, we have transcription. And so something to note about cccDNA, I'll just go ahead and put it over here. cccDNA details. Is the cccDNA contains two sequences called direct repeat 1 and direct repeat 2. So DR1 and DR2. All right, so the RNA polymerase begins transcribing the RNA. I'll put steps here. So RNA pol 2 transcribes the RNA ahead of transcribing the DR1 region of the genome. And this RNA, is called pgRNA, or pregenomic, meaning it's being transcribed before the genome is being formed. All right, and the second step of this is the negative sense is transcribed first until the DR2 region is passed, and then the DR1 region is looped. And so remember how we have, this is a covalently closed chromosome. And somewhere on here we have the DR1 and the DR2. And so the negative sense is going to be transcribed first until the DR2 region is passed and the DR1 region is looped. That makes it where there are copies of DR1 at each end of the newly formed strand. All right. So part of the strand forms the epsilon fold. And this is where the P protein, which is the reverse transcriptase, binds. And this, you might be wondering where this came from. This is what was just translated. And so the P protein binds, or it reverse transcribes a small portion of the strand, forming a primer. And that is going to bind the complementary strand. All right, the P protein then reverse transcribes, so reverse transcription of the negative sense DNA. negative sense DNA portion of the genome. And the extra portions are degraded by the enzyme RNase H. And remember we talked about RNase H, let me find it, up here, and reverse transcriptase. It's one of the functions of reverse transcriptase, degrades portions of nucleic acids. And we don't really see that when we're talking about the RNA retroviruses, but that really comes into play when we talk about the DNA retroviruses. All right. So all that's left after the RNase H degrades is the DR1 site. And that is where synthesis is going to start for the rest of the strand. And so we have the sixth step is where the leftover portion of the RNA segment jumps to the other end of the negative sense DNA strand, and it's going to bind to the DR2 sequence. So, binding of DR2 by what is left of the RNA segment. And from there, the P protein will synthesize the rest of the strand. And what it's synthesizing from is really what has just been created. So everything that was originally there is gone now and has been transcribed and then degraded, and now it's what was transcribed is being transcribed again to form the positive sense. And so the genome is then pretty much complete, and it's going to form a full circle because the two strands are complementary. And the P protein at that point is going to finish the positive sense of DNA. So then the pgRNA, which, as we said up here, was the pregenomic. At that point, the pgRNA is going to assemble into the nucleocapsid. So, kind of really haven't given myself much room over here. But right here is that whole process we just talked about, is transcription. Into the pgRNA. And then, sorry, this is a little messy. We have assembly. All right. So that is the full process of replication. Starting up here, we have the different steps of the general replication. And then going back over here, remember how we went through each one of these, and you can go rewind or, take a picture of the video right here or pause it, and you can see how each of these compares to the other one side by side. So I hope this video was informative and that y'all do well with virology.